All right, let's begin. Today, we're going to start our exploration of immunology by first going back in time to establish its historical foundations. After that, we'll begin building a comprehensive lexicon, a vocabulary of the essential terms that you'll need to master to understand this field.
So here's our roadmap. We're going to structure our discussion into 5 main parts. We'll kick things off with the historical origins, the very birth of cellular immunology. Then we'll take a really deep dive into one of the most fundamental processes, phagocytosis. From there, we'll start building our language, defining the core concepts, then identifying the key cells and molecules involved, and finally, putting it all together to understand the fundamental responses and processes that define immunity.
To really appreciate the origins of cellular immunology, we have to transport ourselves back to the late 19th century. This was an absolutely revolutionary time in science, a period that was profoundly shaped by the work of giants like Louis Pasteur and Robert Koch. Now, their research collectively established what we now call the germ theory of disease. They proved, irrefutably, that specific microorganisms were the direct cause of specific infectious diseases. This was a massive paradigm shift, and it created a brand new, urgent question for the scientific world. If these tiny germs are what cause disease, then how on earth does a host, an organism, defend itself against these microscopic invaders? And that very question set the stage for the birth of immunology. It was right in the middle of this exciting environment that a Russian scientist named Ilya Mechnikov made an observation that would change everything. The year was 1882. He was studying starfish larvae in Sicily, and he had this sudden moment of insight. Now, his choice of animal here was absolutely critical. You see, the larvae of these starfish are almost perfectly transparent, and that transparency gave him a unique window, a way to see inside the living animal and watch its internal cells moving around in real time, without having to dissect or harm it. What he noticed were these motile amoeba-like cells just wandering freely inside the larva's body. And seeing this sparked a revolutionary hypothesis. Maybe these mobile cells weren't just passive bystanders. Maybe they were active participants in the organism's defense.
So, Mechnikov's thought process followed the classic scientific method perfectly. First, the observation of these motile cells. This led him to step 2, forming a testable hypothesis, that these very cells actively protect the organism from harmful intruders. To test this, he came up with a brilliantly simple experiment. For step 3, he took a tiny rose thorn and slipped it under the larva's skin. Now, why a rose thorn? Well, it was the perfect stand-in for an invading microbe. It was a foreign body, but it was sterile. His logic was, if my hypothesis is correct, the body's defensive cells should recognize this thorn as something that doesn't belong and mount a response. And that led to step 4, confirmation. The next morning, he looked at the larva under his microscope and saw that the motile cells had indeed moved toward the thorn. They had swarmed it, aggregated all around it, as if they were trying to wall it off from the rest of the body. This was it, the very first direct experimental proof of a cellular defense mechanism.
So based on this elegant experiment, Mechnikov reasoned that there must be these specialized cells inside an animal whose job it is to perform this defensive action. He needed a name for them, so he coined the term phagocytes. He combined 2 Greek words, phagein, which means to eat, and kaitos, which means cell. So a phagocyte is literally an eating cell. He proposed that this process of cells eating things was the primary way the body defends itself, and he called the process phagocytosis. To study it even further, he found another transparent model organism, the water flea Daphnia. He can infect these tiny creatures with yeast and then watch, in real time, as their phagocytes ingested and destroyed the invading fungal cells. This just added more and more evidence to his theory of cellular immunity. Okay, so now that we've established the historical discovery of phagocytes and the whole concept of phagocytosis, let's transition to a much more detailed, mechanistic look at the process itself. You have to understand that phagocytosis isn't just one single event. It's a highly orchestrated, multi-step cellular program, and it's absolutely fundamental to our innate immunity.
The entire process of phagocytosis unfolds in 7 distinct sequential steps. Imagine our phagocyte, and over here, a bacterium. The first step is chemotaxis. This is where the phagocyte is chemically drawn toward the site of invasion. Think of it like the cell following a chemical scent trail left by the bacterium or by damaged host tissues. Once it arrives, we move to step 2, attachment. This isn't just a random bump. It's a specific molecular binding. Receptors on the phagocyte surface lock onto molecules on the bacterium's surface. This attachment is the trigger for step 3, ingestion. The phagocyte cell membrane begins to extend these arm-like projections, called pseudopods, that reach out and engulf the microbe, eventually enclosing it in a vesicle inside the cell. We call this vesicle a phagosome.
Now the bacterium is trapped inside. Step 4 is phagolysosome formation. Inside the cell, the phagosome moves deeper and fuses with another organelle called a lysosome. Now, a lysosome is basically a bag full of potent digestive enzymes. When they fuse, they create a new compartment, the phagolysosome. This fusion kicks off step 5, intracellular killing. Inside that phagolysosome, the bacterium is subjected to a brutal chemical assault. It's hit with a cocktail of enzymes and toxic oxygen compounds, like hydrogen peroxide, that just tear it apart. After the microbe is killed, we have step 6, breakdown. The enzymes continue to work, dismantling the dead bacterium into its basic molecular components, amino acids, fatty acids, and so on. Finally, step 7 is exocytosis. The phagocyte expels any indigestible waste material out of the cell. It's an incredibly elegant and efficient system for neutralizing threats.
All right, so we've just gone very deep on a specific cellular mechanism. Now what we're going to do is shift our focus. We're going to pull back and look at the broader conceptual framework of the entire field. To really understand immunology, you first have to become fluent in its specific language. So this section is all about defining the foundational concepts that form the basis for everything else we'll discuss.
So, let's start with the big one, the definition of the field itself. What is immunology? Well, at its most basic level, it's the study of immunity. And that means studying all the mechanisms of protection against infectious agents, things like bacteria, viruses, fungi, and parasites. But the scope of immunology is actually much, much broader than that. It also includes the study of how our bodies respond to non-infectious foreign substances. And just as importantly, immunology investigates what happens when the system goes wrong. This leads to conditions like allergies, which we call hypersensitivities, or autoimmune diseases, where the immune system tragically makes a mistake and attacks the body's own healthy tissues. The immune system is broadly divided into two major functional arms. Think of them as two branches of a military. First up is innate immunity. The key features here are that it's nonspecific. It recognizes general patterns that are common to many pathogens, not unique molecules on one specific bug. It is not acquired or affected by prior contact, so its response to a strep throat infection is the same on the first day of kindergarten as it is in your 20s. And critically, it is not mediated by lymphocytes. The phagocytosis we just discussed, that's a classic example of innate immunity. Now, in sharp contrast, we have the second arm, adaptive immunity. This system is highly specific. It targets unique molecules on one particular pathogen. It's a system that develops after you've had contact with an agent, meaning you have to acquire it. It is mediated by lymphocytes, and its most defining and powerful characteristic is immunological memory, which allows for a much faster and stronger response the second time you see the same pathogen.
Central to this whole idea of adaptive immunity is the concept of the antigen. An antigen is defined as any molecule that can react specifically with a component of the adaptive immune system, like an antibody or a lymphocyte. Now, a very closely related term is immunogen, and this distinction is absolutely crucial. An immunogen is an antigen that is capable of inducing an immune response. So all immunogens are antigens, but not all antigens are immunogens. Some small molecules, for example, can be bound by an antibody, but can't trigger the response on their own. Now let's zoom in even closer on that antigen molecule. An antibody doesn't recognize the whole thing. Instead, it recognizes a very specific, small part of it. This precise binding site is called the antigenic determinant, or more commonly, the epitope. A single large antigen, like a protein on a virus, can have many different epitopes on its surface, each capable of being recognized by a different antibody.
So, when our bodies encounter an antigen, the outcome can lead to one of two very different states. The first, of course, is immunity, the state of protection that we've been talking about. But the second, and equally important, state is immunological tolerance. Tolerance is a state of specific unresponsiveness. It's an active process where the immune system essentially learns to ignore a particular antigen. And it's important to realize this isn't a failure of the system. Tolerance is a vital, carefully regulated function. The most critical example is tolerance to our own self-antigens. The immune system has to be able to tell the difference between the body's own molecules and foreign ones. Tolerance is the mechanism that prevents it from launching a devastating attack against itself, which is exactly what happens in autoimmune disease. Okay, now that we've established some of these core concepts, let's move on and examine the specific cellular and molecular players that actually do the work. We're going to look at where these cells come from and what sorts of molecules they use to communicate and to carry out their functions. So where do all these immune cells come from? Well, all the cells in your blood, red cells, platelets, and every type of immune cell, all originate from a single common source, and that source is the hematopoietic stem cell.
These are remarkable multipotent stem cells that live inside our bone marrow. That's the soft, spongy material found in the cavities of our bones. These stem cells can differentiate down various pathways to create all the different blood cells. A key pathway for us is the lymphoid lineage, which produces the lymphocyte. The lymphocyte is really the central cell of adaptive immunity. If you look at one under a microscope, it's a small, round white blood cell, and its most obvious feature is a very large nucleus that takes up most of the cell's volume.
Now, there are two major classes, or subsets, of lymphocytes. We call them B cells and T cells. As you can see, both of them originate from those hematopoietic stem cells in the bone marrow. But, and this is a critical difference, their sites of maturation are different. B cells, or B lymphocytes, complete their entire maturation process right there in the bone marrow. Their primary job in an immune response is to differentiate into what we call plasma cells, and these plasma cells are the factories responsible for producing and secreting antibodies. T cells, on the other hand, also start in the bone marrow, but they then migrate to a specialized organ in the chest called the thymus to undergo their maturation. Their main role is to orchestrate and carry out what we call cell-mediated immunity.
T cells themselves are not all one and the same. We can subdivide them even further based on specific protein molecules they have on their cell surface. We call these molecules cluster of differentiation antigens, or just CD antigens for short. Two of the most important ones to know are CD4 and CD8. T cells that express the CD4 molecule on their surface are known as CD4 lymphocytes, and they primarily function as T helper cells. Think of them as the generals of the immune response. Their job is to help activate and coordinate the actions of other immune cells. Then you have T cells that express the CD8 molecule. These are known as CD8 cells. This population includes the T cytotoxic cells, which are the soldiers. They are capable of directly finding and killing host cells that have been infected with a virus or have become cancerous.
So let's turn our attention now to the key molecular weapon of that B cell lineage, the antibody. An antibody, which is also known as an immunoglobulin, is a glycoprotein molecule. It is produced and secreted by plasma cells, which, remember, come from activated B cells, and it's made in response to a specific immunogen. This really highlights a core principle of adaptive immunity. An antibody molecule will react specifically and only with the substance that triggered its formation in the first place.
The structure of an antibody is perfectly suited to its function. The basic unit is called a monomer, which is a Y-shaped molecule made of 4 protein chains. Right at the very tips of each arm of that Y are the antigen binding sites. These regions are what we call hypervariable. That means their amino acid sequence is incredibly diverse from one antibody to the next, which is what allows our immune system to generate antibodies that can recognize a virtually infinite number of different epitopes. Each monomer has 2 of these identical antigen binding sites. The entire arm of the antibody that contains one of these sites is referred to as the Fab region, which is short for fragment antigen binding. Now, while many antibodies exist as these single Y-shaped monomers, some can link up. For example, the antibody IgM forms a pentamer, which is a large complex made of 5 of these monomer units all joined together.
The final molecular player we'll define is the complement system. Now, it's very important to understand that complement is not a single molecule. It's a complex system of over 20 different proteins that are found in our blood serum. Normally, they just circulate in an inactive state. But when they get activated, usually by an antigen-antibody complex or directly by the surface of a pathogen, they trigger a sequential cascade. Think of it like a chain of dominoes. One protein activates the next, which in turn activates the next. And this leads to a massive amplification of the initial signal. The biological effects of this cascade are incredibly diverse and potent. They help promote inflammation, they enhance phagocytosis by coating pathogens, and they can even culminate in the formation of something called a membrane attack complex, which literally punches holes in target cells like bacteria, causing them to lyse or burst.
All right, in this final section, we're going to start to integrate everything. We'll take the concepts, the cells, and the molecules that we've just defined, and we'll see how these components all work together to produce coordinated immune responses and complex biological processes. We mentioned that the adaptive immune system has memory, and this is perfectly reflected when you compare 2 distinct types of response. The primary immune response is what happens the very first time your body is exposed to a specific antigen. There's usually a bit of a lag phase while the right lymphocytes are found, activated, and start to multiply, and the overall response is generally of a lower magnitude.
In contrast, the amnestic response, also called the secondary or memory response, is what happens upon a second or third or any subsequent exposure to that exact same antigen. And this response is much faster, much greater in magnitude, and far more effective than the primary response. This enhancement is the direct result of immunological memory, where you have a pool of long-lived memory B and T cells just waiting, ready to respond immediately. This, right here, is the fundamental principle behind how vaccines work.
We said earlier that T cells are responsible for cell-mediated immunity, or CMI. So let's formally define what that means. CMI includes all immune responses that are mediated directly by T lymphocytes themselves, rather than by antibodies that are circulating in the body's fluids or humors. Cell-mediated immunity is absolutely critical for dealing with intracellular pathogens, things like viruses or certain bacteria that actually hide inside our own cells. Antibodies can't get in there to fight them, so that's where CMI comes in. T cytotoxic cells can directly kill these infected host cells, and T helper cells can activate other cells like macrophages to make them better killers.
Now, let's look at a process that is just a brilliant illustration of how the innate and adaptive immune systems cooperate. It's called antibody-dependent cellular cytotoxicity, or ADCC. It's a really fascinating mechanism. Here's how it works. It starts with an antibody, a product of the adaptive system, which specifically binds to an antigen on the surface of a target cell, like a virus-infected cell. Now, an innate effector cell, for example, a natural killer cell, comes into play. These NK cells have special receptors on their surface called Fc receptors. These receptors are designed to grab onto the constant region, or the stem of the Y-shaped antibody molecule. So the antibody acts as a bridge. Its antigen-binding arms grab the target cell, and its stem is grabbed by the NK cell. This binding activates the NK cell, which then unleashes its deadly payload of cytotoxic granules to kill the target cell. So you have a non-specific killing mechanism delivered with the absolute specificity of the adaptive immune system. It's beautiful teamwork.
Finally, we have to understand the fundamental process that kicks off most adaptive immune responses: antigen presentation. The thing about T cells, especially those CD4 T helper cells, is that they can't recognize an intact antigen just floating around. They need to have the antigen presented to them in a very specific way. This is the job of specialized cells called antigen-presenting cells, or APCs. These include cells like macrophages, dendritic cells, and even B cells. What an APC does is it first ingests a foreign entity, like a bacterium. Inside the cell, that antigen is partially degraded, chopped up into small peptide fragments. These fragments are then loaded onto special display molecules, and that whole complex is moved to the APC's outer membrane, where it's put on display. It is this presentation of that specific peptide fragment that can be recognized by the receptor on a CD4 T helper cell. That recognition event is the essential first step that activates the T helper cell, allowing it to then go on and orchestrate the entire adaptive immune response.